Okay, we're going to finally cover the process of genetic exchange called transduction. Transduction is the third way that genetic exchange can occur between different bacteria cells. And so this process actually relies on bacteriaphages. So bacteriaphages are viruses that have bacteria as their host. So, we're of course familiar with the coronavirus. Coronavirus is able to infect human cells. And so these viruses that we're interested in instead are able to target bacteria cells, take them over, and then use them as a means to reproduce. So in this process, it is possible for different bacteria cells to transfer DNA from one cell to another, from a recipient cell to another. And so if this occurs, this is referred to as a process called transduction. And so here we see a bacteriophage that targets E. coli bacteria. So T4 phage is a very famous phage that was studied, 50 years ago. And so the way it looks is, kind of looks like a very creepy face hugger from Alien. It looks exactly what you would think like a phage would look like in order to infect something. And so this is the particle, the entire infecting phage particle. And so it has a head part, and so the head part is where the DNA genome is kept. So this is a linear strand of DNA, and it encodes a large number of factors necessary either for taking over the bacteria cell or for producing new phage particles. And then it also has this core and sheath, and then also the fibers that are used to attach to the bacteria cell. So if this phage particle finds itself floating through water, floating through whatever, and then it comes into contact with a bacteria cell that it can actually infect, these fibers are able to bind to the cell wall, and then once that occurs, it's able to inject DNA through the core and the sheath into the cell. So all this DNA is simply enough, and because it encodes a large number of factors, and it also includes DNA sequence that allows for the host machinery to actually convert the DNA into RNA, so it can transcribe that DNA into RNA, and then that RNA can be translated by ribosomes in order to produce proteins from that viral DNA. And so one of the most important things that is produced for the virus are proteins that are able to inhibit the cell and actually kill the cell. So here we just see some EM pictures of phages as they're attaching to a bacteria cell wall. So here are 3 phages that have attached to the bacteria cell wall. One of them has not injected the DNA yet. One of them has injected the entire contents of the DNA. So this is often referred to as ghosts. So these are phage particles that no longer have DNA within them. And then here we see a phage caught in the middle. So some of the DNA has actually transferred into the cell, but we still see that there's a decent amount of DNA remaining to be transferred through. So what is the life cycle of the phage? So if a phage actually is lucky enough to find a host bacteria, it binds to and attaches to the cell wall of the bacteria, and then that DNA is actually injected into the host. And so here we see the phage DNA in red that is injected. That phage DNA is transcribed into mRNA. That mRNA is assembled into new proteins. So if there's multiple genes encoded. And so one of the jobs of one of the genes is actually to degrade the host chromosome. And so that kills the bacteria cell and prevents it from mediating a response to try to fight off that phage. Now it's able to take over the entire host ribosomes and ribosomes and polymerases, RNA polymerases, and it is able to produce the factors that allow it to replicate. So where replication occurs is within the cell. So you have to replicate both the genome. So you'll see a large number of new DNA copies of the viral genome are produced by DNA polymerases, and then you'll also see a large number of proteins are produced that assemble into new viral particles. So these are kind of built in a modular way. So some of the proteins assemble into the head, some of the proteins assemble into the tail, and eventually all of this must come together to produce mature phages. So these mature phages assemble all the different parts, and then they also have a single copy of the viral genome that has been placed within the head of the virus. Once enough of these particles are produced, the host cell is lysed, and now you've released these phages back into the environment where they can find new host cells. And so, a fraction of these will be lucky enough to find a new bacteria host cell. So this obviously seems very bad for the bacteria. It doesn't seem like this is something that they would want to have happen, but it actually is a process that can be used to exchange bacterial DNA from one cell to another. And this is very famously shown in an experiment called the Lederberg-Zinder experiment. They used salmonella, and the salmonella strains that they used were oxytrophic. So just as a reminder, that means that they're unable to grow on minimal media. And so the setup of this experiment was to create a tube, and this tube contained media, and this media was separated by a single filter. And so this filter prevents this filter prevents bacteria, individual bacteria cells, from moving back and forth. And so we've restricted this strain right here to this side of the tube, and we've restricted this strain called LA22 to this side of the tube. And so these strains have different genotypes, but they're both auxotrophs. So this is an auxotroph because it's unable to produce methionine, it's unable to produce histidine, and because of that, it has to be supplemented. The media has to be supplemented with those amino acids. Strain LA22 is unable to produce phenylalanine. It's unable to produce tryptophan. It's phe minus, trip minus, and so you have to supplement those amino acids within the media for this strain to live. And so what they found was that they could take bacteria in this setup, so this filter allows media to move back and forth between these two tubes. And what the experimenters found was that if they took if they took a sample of this culture and plated it, they were unable to grow any sort of colonies. And so no prototrophs were able to develop from this experiment. But if they took strains from this side, if they took bacteria from this side, from the strain LA22 side, they now were able to plate these bacteria on minimal media and incubate, and they found that a small amount of bacteria were able to grow. And so that indicates that there's been some sort of genetic exchange to produce these new prototrophs that have wild-type copies of all 4 genes. So neither one of the starting strains had wild-type copies of all 4 genes. Both of them had 2 wild-type copies and two mutants, but somehow in this process, genetic exchange was able to occur so that we now have a strain that has all 4 wild-type genes. And so how is this possible? And this is possible because there was actually a phage infection that had occurred within this experiment as well. And so this genetic exchange relies on the fact that there's a defective phage production is not perfect. So the goal is to produce a large number of viral particles that contain viral genome, but errors occur, and sometimes bacterial DNA is packaged into the new phage particles. And so, we see this life cycle. The phage infects the cell and injects the phage DNA. This DNA is transcribed and translated into proteins. Again, the host genome is degraded. The viral DNA is reproduced to create a large number of copies. Phage protein components are produced, and they begin to be assembled into mature phage particles. But at some sort of rate, instead of packaging up the viral genome, instead what ends up getting packaged up into the viral particle is a piece of the original host DNA. So this is bacterial DNA that encodes bacterial genes that gets injected into this particle. So again, we have release because the cell is lysed, and if this particular particle is lucky enough to find a bacterial cell, again, you've injected that DNA within the bacteria host. But now, instead of the cell dying, this DNA doesn't contain any of the viral factors necessary for killing the cell. And so instead, all you have is bacterial DNA, and so that can actually be integrated into the recipient chromosome, especially through recombination. And so if this piece of DNA is homologous to part of the bacterial chromosome, then you can have recombination occur. where you have genetic exchange. And so this allele can be replaced by whatever allele is present in this donor DNA. And so the idea behind this experiment is that these bacteria underwent of infection, and so at a small rate, viral particles were produced that had the wild type alleles of these two genes, and then the viruses were able to pass across this filter, and so they were able to actually infect these bacteria cells and supply them with wild type alleles that were then integrated into the host genome. And so you've converted these genes from the defective versions into the wild type versions. And so this is kind of the way that you were able to produce this new strain that had wild type copies of all 4 genes, and so could now grow on minimal media.